All right, guys, I'll try not to make this too long and do things kind of quickly. This is a picture of what I had on the board in class that I was going to go over. And it just kind of summarizes. I like to do this kind of sort of in an outline list, all the things from worksheet number 1 and worksheet number 2 that I want to make sure you know that are important for the quiz. And a lot of this stuff, then I'll quickly go over a couple of things on the answer keys that I just want to point out that people get wrong, because AP is very picky about some things. So I divided it with the pink line on the left. Things 1 through 4 were things covered on worksheet number 1. So let me talk about that first, just kind of an overview. And this thing, does this thing let me point? Oh, good, it does. Okay. So I think everyone's done number 1 before. So the independent variable is what you decide what you're testing, and the dependent variable is what you're going to measure in response to that. And then the controlled variables are everything else that you're going to keep the same. So if you're testing different colored lights on plant growth, you have different colored lights. The growth or the height, let's say, of the plant is your dependent variable. And then you have to think of everything else, that you want to keep the same. Same pot, the same soil, the same water, and everything. And this is very important when you guys are writing a lab report and you're writing your procedure, that you clearly think of all these things. So I think most people are good with that, choosing the independent and dependent variable. Hypothesis. I think everyone knows what a hypothesis is, but AP is a little different, and I think a lot of people are kind of confused by null hypothesis, alternative hypothesis, experimental hypothesis. So just to kind of sum it up, alternative and experimental hypothesis are pretty much the same thing. They're both saying that there is going to be a difference in the groups, like in your dependent variable. Like that, basically these two are saying, these two are going to say that there is a difference. The independent variable is causing a difference in the dependent variable. The only difference is that the experimental hypothesis is how we write it for an experiment, where we write it as an if-then. And a lot of people have trouble with that. And, I say just try to think of it, it's kind of like if and then. And just remember, you're saying in this one, you're basically saying your independent variable. So if my independent variable causes whatever I want to say in my dependent variable, then, and this part's pretty much always the same after the then, you say there will be a significant difference. It'll almost always say a significant or statistical. Those two words mean the same thing. But you don't just want to say there will or there won't be a difference, because there's always going to be a difference. We always kind of say there will be a significant or statistical difference in whatever you're testing. Like if we're doing different colored lights, you say if different colored lights cause plants to grow taller, then there will be a significant difference in height between the plants grown at different colored lights. So that's basically it. A lot of people have trouble with that. A lot of people flip it and they write the dependent variable here. They say like, if the plants grow different heights, then light causes different color growth. So it's just kind of get used to, there's several examples in the homework. So just kind of look at my examples in my answer keys and kind of try to kind of get that pattern. The alternative hypothesis is really kind of the same thing, but it's not written as an if-then. So like the way we might set, like the alternative hypothesis might be stated like that, and the null would be stated like that. If we're doing a test, like we're going to do a test called the chi-square. See this little x squared? It's like a Greek letter. A chi-square is a statistical test that we will do that, gosh darn it, that will determine if there is and so here you'll notice I say a significant difference. You can say significant or statistical difference. You don't have to decide if the difference is significant or statistical. We'll do either the chi-square test, or like you already learned, if you're taking the averages of your groups and the means and the standard deviations and you're doing error bars on a graph, you can use error bar overlap to determine the answer. But this says here only one of these is true. Okay, so only one is true. Either the null, just think of the null hypothesis of no difference. Either there's no difference between your groups, no significant difference after you test it, or there is a significant difference between your groups. And the answer for those two is not subjective. You'll either do a chi-square or use error bar overlap to tell you which. So a lot of times when we're doing this, just the statistical test, like a chi-square, it'll say write the null and the alternative. And you don't have to write it as an if-then for that. You just write it like that. There's not a difference and there is a difference. But if you're actually designing an experiment, it's going to ask you, like, what is your experimental hypothesis that you're testing? Then you write it like this. And so your if-then statement is always saying, like pretty much that there's going to be a difference. And then the null is the other. Now, when we make conclusions, and you're probably not used to this, when you make conclusions, instead of, let's say we test different colored lights on plants, and there is a statistical difference, okay? Let's say the ones in green light or blue light grow a lot more than the ones in green light, okay? Instead of saying blue light causes more growth in plants, they want you to get away from saying that word cause, because you really don't know. All we know is that the blue light one grew statistically more than the red light. We're not sure exactly that's a cause. We're kind of thinking that might be, but the way they want you to make a conclusion is to just use these terms. You either say, we accept the null, which basically means my independent variable did not cause a difference. Or if it looks like there is a significant difference in your data, after you do a chi-square or an error bar overlap, instead of saying my independent variable caused my dependent variable, you just say in your conclusion, we reject the null. And by rejecting the null, you're saying that there was a difference, and that's all we're saying. We got to be really careful about saying I proved this, or we know that this caused that. The words prove and cause are kind of bad kind of buzzwords, okay? And so that's kind of one that's kind of confusing, I think, in the way AP Bio does it. It's maybe different. So just make sure you kind of know. The null means there's no difference. You're trying to prove that or disprove that. And then I think everyone's good with the control group versus the experimental groups. The experimental groups, you're testing your independent variable on it. You might have one, you might have many, depending on how you designed your experiment. And then you should have at least one control group. Often, good experiments have more than one control group. But if you're designing an experiment for me and there's no control group, or for AP, you'll lose points for not having at least one control group. And so sometimes people have trouble with this because depending on the scenario, the control group, how you think of it's kind of different. Think of it either as like the normal condition for that organism you're testing in that variable, or think of it like if I have a group where I just remove the independent variable. Now that's not always easy. So let's say you're doing an example where you're testing different temperatures on plant growth. Let's say your independent variable is temperature. Well, you can't really do this one. You can't remove the independent variable. You can't have a group where you remove temperature and have no temperature, okay? So in that case, try to think of what the normal condition would be. Like maybe you would look up for that plant, what are normal conditions, temperatures that it lives at, or sometimes we just do like room temperature is always the normal condition for temperature. If you're testing, let's say, different types of fertilizers in your experimental groups, then your control group is easy to remove it. You could just have a group of plants with no fertilizer. So think of like those two as sort of your options to help you think of a control group. And then there's also negative controls, which these are ones where you're removing a condition or like a component, let's say, of a test tube that you're putting in there, and you're validating your method. And we'll see an example with that in worksheet 1. And I did an example too in the notes where you like if you're putting enzyme and substrate in the tube, and we're assuming that it's going to make bubbles to show us how much product is being formed. You make a tube that's missing enzyme, and then you make another tube that's missing substrate, and you shouldn't get bubbles, and that just validates your method. You're saying that bubbles are the product being formed, but what if they're not? And a lot of times in AP Bio labs, the negative controls are kind of already set up for you, but you'll see, like when we're doing an experiment, we'll have like, one bacteria plate that's the real experiment, and then there's all these other ones where they're just removing one thing just to validate their negative controls, just to kind of compare and make sure that what we're saying we're doing is, what we're doing. Sample size, this is pretty easy. Like the larger sample size is better. There is no the only magic number, like somebody asked in my first period class, well, what's the cutoff? Like, because there's a question about the Olestra chips, like, their sample size was 1,100, and I asked you, is that large enough? And there's no right answer to that. I mean, yes, it's large enough. The real minimum is 5. Because you have to have at least 5 in each group in order to take an average or a mean. Statistically, they say you should have at least 5 numbers in each group, or 5 things in each group. So 5 is the absolute minimum. If you design an experiment and you had only 2 in there, I would say, okay, that's not enough. But 5 is great, but 10 is better, 20 is better, 100 is better. Don't say like, I'm going to test 1 million plants in each group, because that's unreasonable. So generally, if you're designing an experiment, they want you, the larger the sample size, the better, but it has to be reasonable. Okay, don't go crazy, and you'll be fine. And in general here, the smaller the sample size, the larger the sampling error. And remember, sampling error is just defined as when you take a small group, if you have a small sample size, like if I only take 2 people out of good counsel, and I measure, and I look at maybe like how tall they are or something, and I assume that the height of those 2 people is representative of the whole population at good counsel. I'm likely to be way off. That's going to have large sampling error because my group was small. But if I take 100 people from good counsel and measure their height and average it, if I have a larger sample size, I'm going to be closer to the real average of the total population. So I'll have a smaller sampling error. So basically, you just want, you want this. You want a larger sample size, and that reduces sampling error. Okay, so all of that was what was important on worksheet number 1. So let me just real quickly point out a couple things, and then you guys can ask me in class anything else. Scientific method, don't memorize the steps. I'm not going to ask you to list the steps of the scientific method on a quiz. I mean, generally, you should kind of understand this is what we're doing. But I'm not going to ask you to list them. That's kind of a stupid question. I added here, okay, so the difference again between your experimental hypothesis, which is an if-then statement, and your null hypothesis. I just added, instead of just saying no difference, you're supposed to say no significant difference or no statistical difference. Because there's always a difference. If I just take 2 groups and measure them, they're never going to be exactly the same average. Hardly ever. I'm assuming everyone's good with the definitions there in number 3. Those are pretty easy. I just talked about them. Down here in number 5, I wrote the word conclusion because what I just talked about with sample size is something you can always say when you're writing a conclusion to a lab report. And we're going to do a full-blown lab, like 2 weeks, where you're going to do all of this stuff. We're going to do bugs. I know that sounds gross, but I think we're going to do pill bugs. But you're going to design an experiment to test something on them, an independent variable. And you're going to do it from beginning to end. Writing your hypotheses, designing the experiment, telling me how, and setting, you're going to actually do it. You're going to set the whole thing up. You're going to do data tables and then graphs and then write a conclusion. And what you often have to do in a conclusion, besides just say, do I accept the null or reject the null, which is basically telling me if your hypothesis was correct or not, it'll ask you, well, what could you have done to improve the experiment? And everybody scratches their head like, I don't know. One thing you can always say, always, and get 2 points just for saying like, well, it would have been better if I, had the materials and the time to get, instead of testing 10 plants in each group, or bugs or whatever, it'd be better if I had 100 bugs in each group. We just didn't have that supplied to us. So you can always say a larger sample size would be better because it'll reduce sampling error. That always gets you a point in the conclusion. And repeating it, just say like, we only did the experiment once in class, but it'd be better in science. You're supposed to be able to do the same thing over and over and over and get the same results. Okay. So here's the Olestra example. And I used to bring in these chips. I used to be able to find them in the store, and I would bring in Pringles with Olestra and Pringles without, and we would taste them. I can't find them anymore, but so just look at number 7 and 8 there. Again, that's usually where people lose points. Again, I'm going to grade yours that you've turned in, but kind of look at how to write the if-then statement for the experimental hypothesis. So if this is my independent variable, Olestra causes cramps, my dependent variable, then the group that eats the Olestra chips will have either, you can say one or other, significantly or statistically more cramps than the other group. That's pretty much kind of always how we state it. The null hypothesis is just that there will be no difference or no significant difference in the amount of cramps between the two groups. Again, here, sample size was 1,100. I asked you to comment on that size. There's no right answer here. I mean, yeah, that's pretty large. That's good. More would be better, but that's pretty reasonably large for, as long as it's not below 5, you're going to say it's pretty good, but more would be better. I'm assuming everybody's good with the experimental group, control group, the variables. I think this is pretty easy. One thing people lose point on in labs and quizzes and whatnot, when I ask you to tell me the controlled variables, so the things you're keeping the same that you're not testing, a lot of people try to just kind of sum it up and they go, okay, my controlled variables are temperature, light, or not, that doesn't apply to this one, but what about temperature? You have to actually say these words. It's going to be in the same temperature, and I'm going to put them in the same test tubes so they're the same material and the same. Don't just say temperature, light, soil, pot. Like, what about the soil? Just say I'm going to use the same type and amount of soil. I'm going to use the same pots or the same size. Be specific here. The only thing you should not have here, and some people do, some people say the chips. Okay, well, you're not specific enough. I don't know what you mean, because the independent variable is the type of chip, right? Our independent variable is the type of chip, whether they're regular chips or chips with Olestra. So if you just say chips as controlled variables, I'm going to mark that wrong. Okay, but here you have to say we have the same, they all had the same amount of chips, like everybody ate 13 ounces of chips. Be very specific in a lab when you're telling me about the things you're controlling. Tell me how you're controlling them, not just they're going to have the same amount of chips. Say they're all going to have 13 ounces of chips. Okay. Number 13, again, there might be more that I didn't list there that you thought of. Whenever you're dealing with living things, especially humans, it's hard to control that. So it's possible, that the group that ate the Olestra chips just happened to maybe have ailments in their stomach. I don't know. That's why the best way to do that is you could say, okay, I'm going to pick people all from the same family, or I'm going to pick all females or all males, and try to take some of the variability out of humans. But the best way to do it is just do a larger sample size and take an average. 14, you see here, I actually drew the bar graph. You didn't have to draw the bars on there, but I just kind of showed you what the graph would look like. If we did this graph, it would be a bar graph because we have discrete data here. The type of chip, that's a category. But all you had to write was the type of chip on the x-axis, because that's my independent variable. There's no unit, so leave that blank. And then I wrote the percent of cramps. You could have wrote the number of cramps here as your dependent variable on the y. That's all you had to write. Then down here it says, suppose we do this chi-square test, and we figure out there's no difference. But the other way, what might happen is maybe you graph the mean and the standard error, and what does it say here? There's no difference. Okay, so maybe you do the error bars, and like that, you see that these error bars overlap. So from that, I could say there's no difference. And the way to say that is we say that the null hypothesis is supported, which means there was no, obviously there was a difference, but it's not considered a significant or statistical enough difference that we say there was a difference. So this was the one, if you read through, where it talks about negative controls. So in each tube, we put the same amount. We put 4 milliliters of hydrogen peroxide. Centimeters cube is the same as milliliters. So we put the same amount of it. So the substrate is peroxide. You might remember enzymes from freshman year. This enzyme catalase takes this substrate and breaks it down into these products. And what we're measuring here is the product oxygen, they're saying, is what these bubbles are. Again, we have to kind of validate that method with some negative controls. But so you'll notice first here, as controlled variables, we have the same tubes, and we put the same amounts of enzyme in each tube and the same amount of substrate in each tube. And then a negative control would be, maybe I show you on the next page, to leave the have another tube where you leave the enzyme out and replace it with water, so the volumes are all still the same. But if the enzyme isn't there, right, you shouldn't get bubbles. But you need to have that tube to show that. And then maybe another tube where you leave the peroxide out and replace it with water to keep the volume the same. And that should not get bubbles. If you got bubbles in any of those negative controls, what that means is that our method, we're saying that these bubbles are the oxygen produced by this reaction. Well, that's not true. Maybe there's something else causing bubbles. Okay, so those would be negative controls, to kind of take the things we're putting in the tube and leave one of them out in each tube. Another control might be to do, okay, what would be normal conditions? Again, with temperature, you're like, oh, I don't know what normal conditions would be. But maybe you look up catalase. Maybe you got this catalase from a potato, and you look up what are the temperatures normally for that potato and where it lives. And maybe you would do a tube under whatever that would be, whatever that normal temp would be. That might be a control group. Okay. Or another one is, I just mentioned, leave out the enzyme, that kind of thing. All right. So you guys can read through that. I already posted it. Again, there's another example of, this is where people usually get points off, writing an if-then experimental hypothesis. Just look through that, and you can ask me questions next class. I will also say this, units are a huge thing. People lose points. I constantly am writing -1/2, units. AP is so picky about that. Anytime you're talking about a variable, if it has a unit, just you put what units you're going to use, okay? Just always write a unit with everything you're talking about. You get points off for that. And let's see, again here in number 21, look how specific I explained the controlled variables. I didn't just say, okay, what am I going to control? The tube, the enzyme, the this, the that. This. I explained exactly how I'm keeping all these things the same, so that the only difference is the temperature. There's a typo there in my answer key. That should have been 60 degrees, not 40. And the rest of this, I kind of just talked about this. These are the negative controls. Again, they're often used to validate our method. So let's say in this tube, you leave out the enzyme and replace it with water. And in this tube, you leave out the H2O2 peroxide and replace it with water. And you should not get bubbles in these. If you do, your whole experiment is poop. Okay? You have to start over. All right, I think the rest is pretty self-explanatory. Okay, so that's number one. let me go back to my picture from the board. Okay, so worksheet number 2 was about these last few things here. I'll just kind of point it out. So data tables, it's really important that your title has the subject, the independent, and the dependent variable in it. And the word mean, if you did the mean. So if the data table is, you took the average or the mean of a group of data, if that's what you're showing me in the table, you have to say that in the title. Okay. If it's raw data that you didn't take the mean or the average, then you don't have to say it. And make sure it's really descriptive. Independent variable always goes there with unit in parentheses. The dependent variable always goes in the next column with unit. And again, you might have multiple columns of the dependent variable depending on if you're measuring it every week or whatever. And make sure your data is all at the same decimal place. I will say significant figures that you probably did in chemistry, and it's kind of a nightmare to figure. Don't worry about that. AP doesn't really care about significant figures. All they care about is that, whatever decimal place you're going to record your data to, it's all the same. And usually on an AP test, they'll say, like, do this and round all your answers to the 10th of a decimal. They'll tell you what to round it to, and don't really worry about significant figures, even though I had that in the yellow pages. Then for data, there's different types. You just need to know that so you know what kind of graph to do. Qualitative will do that with every lab, where you'll just write a little paragraph where you describe what you're seeing and stuff. But you should design every experiment. You should design when you're designing an experiment, you design it so that your dependent variable is a number. So I can't say like, okay, I'm going to test, I want to test how light affects the movement of wood lice. Well, how are you going to, the movement of wood lice would be your dependent variable. Or are you just going to describe them? You can't. Your dependent variable, you have to try to find a way. Maybe you measure the movement of them, like you say, okay, I'm going to measure how many times they move in a minute. That's a number. Or how far they move from their home source, or that would be a distance. So you have to try to design your experiments, and again, we're doing this in 2 weeks, so that whatever your dependent variable is you're going to measure, make sure there's a quantitative way to measure that. So because you have to be able to do graphs and statistics with it. And then I think everybody's good with, we did this freshman year. Discrete data is data that's in categories, like not even numbers, like different colors, different rivers, whatever. Or you're counting something, like you're counting how many squirrels are in a forest, and you can't get a decimal or a fraction. Continuous data is things that, and I underlined the word, can be decimals or fractions, even if you're only recording it to the whole number. So things like, time is always continuous, even if you're recording it like 1 second, 2 second, 3 second. Just by nature, time measurements, any kind of distance measurements, volume measurements are always continuous data. And then the general rule, the graph thing is kind of confusing. I tried to simplify it here a little bit. Because there's lots of pages in the yellow pages, and it's like, well, if the independent's this and the dependent, bottom line, kind of think about what your independent variable is, if it's discrete or continuous. There's really kind of 3 categories. If your independent variable that you're testing is discrete, either categories or counts, probably that's going to be a bar graph. If your independent variable is continuous, it's going to be a line graph. And then sometimes we kind of group these together and refer to the, oops, why is it doing that? It's not letting me write. And call that a line graph, okay? But a line graph could be like you connect the dots and kind of show the curve, or if it's showing a correlation, like if all the points are going up, it's a scatter plot. But a lot of times you won't know which one it's going to be until you plot the points. So I just kind of refer to all of these as a line. Let's see. So sometimes a line graph could be either of those two. And to do either of these, your dependent variable has to be continuous as well, technically. Although sometimes you'll see it's not. But again, there's always exceptions, which make it confusing. And then the other scenario is if your data is in class size intervals, or bins, they call them, like you're measuring, you have like how many people are, or let's do plant. How many plants have between 5 and 10 flowers? How many plants have 10 to 15 flowers? How many? So then you do a histogram where it's like a bar graph, but they're touching. Now, in class size intervals, this could be continuous or discrete. So, for example, if I'm counting how many flowers are on plants and I'm doing something like this where, you know, if you make this bigger, like, you know, like the first bar is how many plants had 5 to 10, and then the second bin is how many plants had 10 to 15 flowers and how many plants had 15 to 20, right? Counting flowers is discrete data, but because they're in class size intervals, you'll do a histogram. Or it could be continuous, where you're measuring like the height of plants, how many plants are 5 to 10 centimeters tall, how many plants are 10 to 15 centimeters tall, etc. So class size intervals can be discrete or continuous. But if you're in class size intervals, you're probably doing a histogram. I don't know what's going on here. All right. I'm going to assume you guys know how to do a graph. There's a lot of that you've done, but the independent variable with unit goes on the X. The dependent variable with unit goes on the Y. Your title has to have all the same things, the independent, dependent, and the subject, the thing you're testing, like you're testing something on mice or whatever. And you have to have an error bar statement, because you should have error bars on most graphs. If you do, you have to tell me do the error bars represent standard deviation, standard error, whatever. We'll learn all those later, but I already talked about error bar overlap. Once you plot the error bars, and we'll learn the statistics that error bars represent later, but if they overlap, there's not a difference. If they don't overlap, then there is a statistical difference. I'm assuming everybody knows how to do the rate or the slope of a line. That's easy. And correlation is pretty easy. There's either positive, negative, or there's no correlation. And remember, correlation is just a correlation. It doesn't mean this caused this. Don't use the word cause in a conclusion. I'll start circling it because you can't for sure know that. So just real quickly looking at I want to look mainly at the graphs. This is worksheet 2. I'm not going to go. I'm assuming you guys all know how to do numbers and discrete versus continuous. That's pretty easy. Again, if there's ones on here, a lot of times people are not sure, well, like, why did you say that should be a histogram? Why is that a bar? Just circle it and ask me, and I can clarify it real quick in class. Like somebody asked me in my other class here, because height is continuous data. So if you put continuous here, that's true. Height is a continuous data. It can be a decimal or fraction. But because we said it's continuous data in class size intervals, that's why I chose histogram. So if you wrote continuous here, you're not wrong. And then again, down here, like line graph, this could be, this kind where you draw a trend line, or a scatter plot where you do a line of best fit. You just won't know until you plot the points and kind of see, does it look like there's a correlation where you do a line of best fit, and a lot of times I'll tell you which one to do. But again, make sure you have units. Like if this was like a test or a quiz, or when I'm grading it, if you didn't have the unit, and this one it didn't give you the unit, so I just put a place for it. But if it gives you the unit, you give me the unit for the independent and dependent variable. Okay, so make sure all of those have units. I think everybody, we haven't really done statistics yet, but in general, I just want you to know a larger error bar. So B had the larger error bar. What that means is that data set, that group that I averaged, is more spread out, and we'll talk about that more later. And group A had the smallest error bar. So that group that I averaged for that data point was less spread out, that had the smallest error bar. All right, so graphing here. Again, units. They were both continuous. Okay, both independent and dependent were continuous. So you should have done a line graph here. All right. Again, you might have had different increments here, but as long as they're all the same, mine go by tens and whatever, these go by twos. My independent variable, unit, dependent variable, unit. You'll lose points for those. Notice my title. I notice a lot of you don't even have titles on it. You have to have a title, and my title has, my independent variable, which is time, my dependent variable, which is the amount of maltose produced, and my subject. That's the thing I'm testing it on. So you have to have at least those three things. In this one, we're not graphing averages or means. So this is just raw data. It hasn't been averaged, so you don't have to say that. And then over here, it kind of got cut off, but I just did the slope of the line to calculate the rate. And you might have got I noticed a lot of people did 0 to 60. It told you specifically the time period 0 to 30. So I took the data points at 30 and 0, and instead of trying to read them off the graph, I highlighted, they're right here. If you have those exact data points in your table, you can just use those exact numbers. So I used these numbers. And that's how I calculated the rate, which is 0.35 micromoles per minute. Whoopsie. Okay, again, units, 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 units. If you have just a bunch of numbers there and no unit, you're going to get a bunch of little -1/2s. Okay, number 16, also, real quick, there was a typo. I think this said standard deviation in the paragraph, and then they gave standard error of the mean in the table. No big deal, but it's standard error of the mean. You don't know what that is yet, probably. But so since my independent variable is categories, it's 2 different rivers. So notice that's in the left column. That's discrete data, so I'm doing a bar graph. Okay, so you should have done a bar graph. Again, the things that you needed to get points for. In your title, your independent variable are the 2 different rivers in Norway. My dependent variable are the number of eggs laid. My subject, it's salmon. And then you need to have the word mean here, because in this one, you're graphing the means. This is an average number of eggs in the river. So you need to tell me that. So you can either have the word mean in the title, or I have it over here in the dependent variable label. You don't have to have both. You have to have it either in the title or on the y-axis, if that's what you're graphing. And you can do both. That's great. And then you must have error bar statement. You have to tell me, the error bars here represent plus or minus 2 standard errors of the mean, which is what they gave you here. Okay, and again, you don't know what that is yet, so that might be a little confusing, but you could also put this, it's possible to just kind of write what the error bars represent kind of in your title, kind of in parentheses. But you have to tell me, if you have error bars on your graph, which for most graphs we will, you have to tell me which statistic you used, because the error bars don't go up and down randomly. They're based on a statistic. Okay, and then you see here the error bars do not overlap. So that means there is a statistical difference between these 2 groups. And so the way that we would say that is we would say we reject the null. That's how the conclusion we would make. There's a statistical difference between the 2 groups, the 2 rivers. So we're rejecting the null, which the null is no difference. So I think that's pretty much what we talked about. That's review all that stuff, and my other class is going to have the quiz right away. I'll give you guys, a few minutes at the beginning of class. If there's anything in here you're still not sure about before we take the quiz next class, I'll give you about 10-15 minutes at the beginning of class if you need it. Or you can always email me tonight, tomorrow, and I can answer you or whatever. It's totally fine. All right, so sorry about that. I know you wanted to have like a day off, but we couldn't afford it.